Hi guys, this is Michael from Bridge Channel, and today we're going to talk about AP Bio Unit 2, Chapter 1, the cell structure. This part will be divided into two parts, and the first one will be about prokaryotes, and the second part will be about eukaryotes. Let's dive in. So to start with, we're going to look at the cell structure of prokaryotes. For prokaryotes, there are two really big characteristics, and the first one would be that prokaryotes do not have any nucleus, and the second one would be that they don't have any membrane-bound organelles. And because they don't have nucleus, their DNA is not inside nucleus. They have something called plasmid, that is a circular DNA inside the prokaryotes, and they are also double layer. Uh, yeah, they are the DNA called plasmid. And the biggest example for prokaryotes would be the bacteria. Let's jump into the cell structures of them. The first one would be the nucleoid. A nucleoid, they don't have something like nucleoid is not nucleus, but it's the place that circular DNA named plasmid exists, and they are not wrapped around protein. The second one would be cytoplasm. Cytoplasm is a gel-like substance and it is a site of metabolism for the prokaryote cells. Ribosome are the parts where they build the protein for during translation. And the plasmid, as I said, they are the small circular DNA inside the cell, and they are double-stranded. Every DNA are double-stranded in prokaryotes and eukaryotes. So if the question asks you that they don't are there like a single-stranded DNA, then it's wrong. And about the plasmid, plasmids can be shared between bacteria, and they contain gene for antibiotic resistance. The next one would be the cell wall. Prokaryotes have a cell wall that provides shape and allow them to withstand the pressure. And yeah, and there's something called Phylly that allows prokaryote cells to attach to surface, or share DNA with other cells, or harpoon DNA from surrounding areas. There's also something called capsule that helps cell from dehydrating and adhere to surface. And the last one would be flagellum and flagella, and they are long extensions used in cell locomotion. They don't really talk specifically about this in AP Bio exam, so you don't really need to know deeply about it, but you have to know that prokaryotes have flagellum and flagella. And they also have cytoskeleton that we're going to explain later in eukaryotic cells. Now let's jump into cell structure of eukaryotes. For eukaryotes, they have four big characteristics. The first one would be that eukaryotes are usually uni or multicellular, and they have nucleus. They have membrane-bound organelles, and they have DNA inside the nucleus which is also double helix or double-stranded. For eukaryotes, it usually contains fungi, plant, animal, and other multicellular organisms, and they are a much more complex type of organism than prokaryotes. So the first organelle of eukaryote would be the nucleus. In nucleus, they control the normal activities. They contain DNA inside chromosome, and they are bounded by a nuclear envelope with pores. And each cell has fixed number of chromosomes inside their nucleus. For example, humans have 46 chromosomes, and if they lack of chromosome, they're gonna have genetic disorder or other symptoms. DNA is not a organelle, but DNA is something important in cell. They are hereditary material inside nucleus, and they store genes. They store gene informations, and we're gonna talk specifically about this in probably Unit 6. The next organ will be a nuclear envelope, which we call nuclear membrane. And they are double membrane. They contain nuclear pores for transportation of materials, and they are connected with the rough ER. Nucleolus is something that's inside nucleus. Is usually 1 to 3 per cell, and they disappear when dividing. The next important organelle will be the cell membrane, or we also call it plasma membrane. And this is a living layer. It means that they function. Uh, yeah, we just call it living layer. And the cell and plasma membrane are always, not usually, they're always double layer of phospholipid and protein, which looks something like this. And there's a protein where materials go in and out, and this is the phospholipid where the head is hydrophilic and the leg is hydrophobic. And plasma membranes are selectively permeable, and this means that they sometimes let the materials in and sometimes they let it out. And this is a really important characteristic since this is how the materials concentration inside the cell are controlled. So right now, you don't need to know really deep about it, but you should know that they are selectively permeable, and we're gonna talk deeply later in this unit. And there's something special about cell membrane of plant. So cell membrane of plant lies immediately inside cell wall, and as you know, cell walls only exist in plants, and it doesn't exist in animals. So yeah, that's a big difference. And the second part is that the cell membrane of plants pushes the wall outside to maintain the shape. If they don't push it outside, then the cell wall will crush them. So that's the important function of cell membrane inside plant cells. The next one will be the cell wall that I talked about a few seconds ago. And for cell walls, only plants and fungi have it, and prokaryotes. And they are usually the rigid and non-living layer of the cell, and they support and protect the cell. They provide the shape, and there are pores that let substance in and out, since cells also need to let materials inside them. This is just just some details. For fungi, their cell walls are made by some substance called chitin, and for bacteria, they are made out of peptidoglycan, and for plants, they are made of cellulose. Cellulose came out in carbohydrates, and it's a way to store their energy. I mean, not store their energy, to form their cell wall. The next one will be the cytoplasm. Cytoplasm provides medium for chemical reaction, which we also call as metabolism, and they provide the shape to the cell, and they contain organelles inside them. And they are usually made of H2O, salt, organic molecule, and other organelles. Cytoskeleton. Cytoskeletons are made of protein, and they maintain the cell shape. And the job that they do is moving organelles around, and they're usually made of two parts, which are microfilaments and microtubules. And microfilaments are made of actin, and they're something that looks like a thread. And microtubules are made of tubulin, and they look something like a tube. Centrioles are paired structure that are located near the nucleus, and they're bundles of microtubules. And they appear during stages of division, which we call mitosis or meiosis. And they form mitotic spindle, and they pull chromosomes apart during division. I'm not going to be really specific about it, but if this is a nucleus and there's chromosomes lined up in the middle of the nucleus, then the centrioles on the polar will pull them over. They're gonna rip the chromosome to each side, and they're gonna divide the cell and go through the mitosis. The next one will be the mitochondria, the plural form of mitochondria, and they are really important. They're the powerhouse of the cell respiration. They create ATP, the energy for us, and they're not in bacteria. And if the cells are more active, they have more mitochondria. And for example, if you're like working out and you have a bunch of muscles, then a bunch of mitochondria will be in your muscle instead of and they're gonna be less mitochondria in other fat cells or something else. And mitochondria are made of double membrane, and they have their own DNA. We're gonna learn this later, but their DNA are maternal, which means that their DNA comes from their mother, and if a child has a genetic disorder in their mitochondria, then it's usually their mother's genetic disorder. Now it's the endoplasmic reticulum that now it's the endoplasmic reticulum that we refer as ER. Sorry, ER are divided into two parts. The first one would be the rough ER, and the second one would be the smooth ER. So what ER does is that they are a network of hollow membrane tubules, and they help move materials around the cell. They connect to nuclear envelope and cell membrane, and what they mainly do is they synthesize cell products and transport them. What rough ER does is that it makes protein for use of outside cell. I mean, not make. They transport and modify the protein. The part that makes protein is the ribosome that is located on the surface of rough ER. In ribosome, they make protein for use of outside cell, and they are usually threaded into a rough ER, and then the protein is transported and modified, and they're moved to other parts of the cell. So the second one would be the smooth ER, and smooth ER is a little bit different from the rough one. They don't have ribosome, but they are attached to the end of the rough ER, and they make lipids instead of proteins. So the important part that you gotta take from here is that the rough ER are related to protein, and they transport and modify protein. And for the smooth ER, you should know that they are related with the lipids. Now let's talk a little more about ribosome. Ribosomes are the ones that make protein and RNA, and they are usually located in rough ER or somewhere in the cytoplasm. And we're gonna learn this later in probably Unit 5 or 6 when we study deeply about the translation and the stages. Yep, I think it was translation. Yeah. And there's something called Golgi structure inside the cell. So Golgi structures are stacks of flattened sacs. It's just like bunch of sacs, and they usually have something like a shipping side and a receiving side, and they receive protein from rough ER, and they transport proteins with vesicles. And vesicles are something like this. They're like something that contains proteins, and these vesicles are located inside the cell, and sometimes they leave or sometimes they join into the cell. So what they do, they usually modify, sort, package molecules from rough ER and transport them. The other one would be the lysosome that is related with the cell death. They contain digestive enzyme and they are programmed for cell death. And because they have digestive enzymes inside them, those enzymes break down and recycle cell parts, and we usually call it as phagocytosis. The next one will be cilia and flagella, and they are made of protein microtubules. They are used for moving cells and fluids or move particles across cell surface. And cilia, they are shorter, but there's a lot of them. Flagella, they are longer, but they have few of them. And the next one will be the vacuoles. The vacuoles are fluid-filled sacs for storage. They just store a bunch of things inside them. And they are usually small or absent in the animal cells, but they are really well developed in the plant cells. There are no vacuoles in prokaryotes and bacteria, and they are usually the storage for protein, sugar, mineral, lipid, waste, H2O enzyme, salt, and yeah, just a bunch of things. And the next one will be the chloroplast, and this only exists in producers, often we call as plants. And they usually they are the place where photosynthesis takes place, and we're gonna learn this later in Unit 4. And they are made of double membrane with outside there's a smooth membrane, and inside there's a modified and the inside membranes are modified into sacs called thylakoid. And they contain their own DNA, and they contain their own enzymes, pigments, and yeah, they don't have this in animals and bacteria. So let's dive deeper into cell membrane for a second. So cell membranes are, we usually call it as fluid mosaics, and we call it fluid mosaics because of the phospholipid bilayer and the mosaic of proteins that are embedded on the cell membrane. And for mosaic of proteins, there are something like integral protein and peripheral protein. And for integral protein, they are permanently attached to the membrane, and for peripheral protein, they're not embedded and they're temporarily attached. You don't need to know really deep about this, but this is really good to know because sometimes they just give you as like a small small sentence in MCQ. These are the list of organelles that are inside eukaryotes and prokaryotes. And if you need this, I'm gonna put this on my YouTube description so you can just download it. And I also have a picture of a cell that I drew, and if you look at it during the lecture, then it's gonna be really helpful. And just a little bit more about the membrane proteins. Their roles are usually receptor, recognition, enzyme, adhesion, channel, pump, and they just do a bunch of things that the bilayers cannot do. And there's something called cholesterol, and in eukaryotes, fat molecules like cholesterol modulate membrane fluidity. So it means that cholesterol determines whether the membranes are like a liquid-ish or like a solid-ish. If it's cold, they are it's more closer to fluid, and if it's hot, they're less close to fluid. That was all for cell structure, and next time we're gonna learn about the cell itself. And yeah, have a great day. Good luck studying cell structure, and please look at the picture because it's gonna be really helpful. Thank you.